Gephyronic acid is a polyketide that exists as an equilibrating mixture of structural isomers. In nature, gephyronic acid is produced by slow-growing myxobacterium, Archangium gephyra strain Ar3895 and Cystobacter violaceus strain CBV76. It is the first antibiotic in myxobacteria that was reported to specifically inhibit eukaryotic protein synthesis. Biological properties. Preliminary studies demonstrated that gephyronic acid inhibited growth of yeast and molds as well as elicited a cytostatic effect through the inhibition of eukaryotic protein synthesis in mammalian cell cultures. Feeding experiments done with radioactive precursors showed a drastic difference in incorporation of leucine by a human leukemic cell like K562, but little difference in the incorporation of uridine and thymidine. This suggested that the primary target of gephyronic acid is protein synthesis. As such, it is a potential target for cancer chemotherapy. Gene expression profiling of human breast cancer cell lines is underway in an effort to further define the potential of gephyronic acid as a chemotherapeutic lead. Screening of a library of compounds derived from myxobacteria found that gephyronic acid was the strongest inhibitor of processing bodies, P-bodies, assembly. P-bodies are discrete cytoplasmic mRNP granules that contain non-translating mRNA and protein from the mRNA decay pathway and from mRNA silencing machinery. Within P-bodies, mRNAs can be degraded, but components of P-bodies can rapidly cycle in and out to return to translation. The mechanism of P-body assembly inhibition by gephyronic acid has not been characterized, but initial studies suggest that the mode of action could be through stalling ribosomes on mRNA or by reflecting early steps of translation initiation, such as binding of ribosomal subunits or initiation factors. The same study also found that gephyronic acid inhibits CIF2-alpha phosphorylation and formation of stress granules under stress conditions. Stress granules contain non-translating mRNAs and translation initiation factors, suggesting that they may form as a result of aggregation of mRNPs stalled during translation initiation. By monitoring immunofluorescence of an established stress granules marker, it was found that stress granule formation was inhibited in the presence of gephyronic acid. Gephyronic acid may have a direct or indirect effect on translation initiation factor EIF2-alpha, which would trap mRNA into non-functional initiation complexes, inhibiting both P-body and stress granule formation. Biosynthesis. Sequencing of the PKS gene cluster in C. violaceus was conducted and confirmed to reveal 5 type 1 polyketide synthases and post-PKS tailoring enzymes with O-methyl transferase and cytochrome P450 monooxygenase. The overall structure correlates well with modular arrangement of PKS-encoded proteins, aside from some unexpected elements that are likely caused by inactive domains. Initial loading uses a GCN5-related N-acetyltransferase (NAT) domain instead of the typical AT domain. Gephyronic acid contains a methyl ether at C5 and a C12C13 epoxide. These functional groups are incorporated by post-PKS tailoring enzymes. GPHA is likely responsible for installation of the C5-methyl ether. O-methyl transferases SPIB and spike used in spirangian biosynthesis exhibit the same SAM binding motif as GPHA. GPHK is a member of the cytochrome P450 superfamily and is suspected to carry out the epoxidation of the C12C13 olefin. Such epoxidation in post-PKS modifications has been seen in apotholone biosynthesis by EPOK. In EPOK, The consensus mechanism of epoxidation by P450 involves the formation of a pi complex between an oxoferryl pi-cation radical species, FeIV, and the olefin pi bond, followed by electron transfer, formation of the olefin pi-cation radical, and finally epoxidation. However, it is also possible that in addition to cytochrome P450, a Fab-dependent monoxygenase is also required to install the epoxide. This codependent process is seen in pterandomycin biosynthesis by TAML. Experiments to clarify the function of these enzymes in gephyronic acid biosynthesis are underway.